So, today, mastering Western blotting optimization, expert tips and techniques for better results. And as Kathy introduced me, I'm coming from Agrisera, and Agrisera is based in Sweden. It is early morning right now here because we're located in Europe, and our winter day looks a little bit like that. If we're lucky, we see northern light. We are quite high up, so in the summer it's mainly a day. So we have winter day and summer night, which is light basically all the time. And our company has been established in 1985, and we have two locations. This beautiful location, which you see on the larger picture, is where our antibody farm is located in the northern Sweden. We are growing quite fast right now. We have 60 employees, became part of Olink Proteomics since year 2020. And here you can see our staff in the facility in Umeå, from where we are shipping the catalog antibodies and also making purification and quality control. Agrisera plant antibodies are our unique brand, and our facility is and our company is a part of Olink Proteomics, and we are providing antibodies for their protein detection platforms. The technology which Olink has developed allows to detect over 1,500 of human proteins in a single blood drop. And our catalog products cover plant antibodies for plant and algae, but we have also quite many of over 100 epitope tag antibodies and high titer secondary antibodies and detection reagents, which are for Western blot and for ELISA. And as we worked with these products for over 20 years, there is a lot of interesting knowledge we can share with you regarding different techniques. And in this case, we focus on Western blotting. And I wonder if you know how old Western blot, how old technique this is. It is allowing to visualize and measure specific protein, unique protein in a complex mix. So think in your head, when do you think the first publication came? 1979. It was used for the first time in two independent laboratories. So you're actually working with a technique which has been around for quite a while. And why is it called Western blot? You're probably familiar with other techniques like Southern blot for DNA, we have Northern blot for RNA, we have Eastern blot for post-translational modifications, and then we have a Western blot for proteins. And actually, Western blot, the name comes from the lab being based on the West Bank, and it somehow fits to the other names. And it is based on the concept of a certain sandwich. Well, I haven't had my breakfast yet, but I hope you're not hungry. The transfer stack in a sandwich, just a reminder how it's looking. We have, of course, I'm talking here about, this is a sandwich for a wet transfer, not a semi-dry. But the principle is the same, that we have proteins which are separated in a polyacrylamide gel, and the proteins are charged negatively by SDS, and we are LDS, and we are applying a current which will move the proteins from the gel to the membrane. And what we are after is, and this is very important here, we are after the exact copy of proteins separated on a gel. We want to obtain an exact copy on a membrane. And who is doing the job of actual visualization of the protein, which is transferred to the membrane. This is an antibody. And just to remind you how this molecule is looking, it's a Y-shaped molecule, and an antibody from a mammal like goat, mouse, or rabbit looks that way. We have so-called constant part, a handle, which is not changing. And we have a part which is important for us, where we have antigen binding domain. And they are also called, this is a variable region, which is called FAB. And the handle is called FC, YFC fragment, which crystallized. A researcher left the preparation in a refrigerator, and it crystallized overnight, and therefore he called it FC part. From the perspective of antibody binding to target protein, that part is most interesting for us. I will remind you, explain how the binding between the antibody and the protein occurs. But just a reminder that we also have antibodies which look a little bit different. You're maybe familiar with them too. They come from llama. As you see, they are smaller. The variable region is different, and therefore these antibodies have it easier to penetrate, for example, in the immunolocalization studies. So antigen binding domains is absolutely crucial, and we're talking here about binding between antibody and our target protein, which we want to visualize. What is an epitope? There are often questions, if we can provide information to which epitope antibody is binding. And that is actually only possible if epitopes are mapped. And this is not a standard practice, which is done, but I will explain how we are choosing the fragment which is used for immunization. We have linear epitopes, which are brought by subsequent amino acids in a linear structure of a protein. So an epitope is from 3 to 15 amino acids long. Okay, antibody can bind to a sequence between 3 to 15 amino acids, and that's a linear epitope. It's very important that you remember that. And it is these epitopes antibodies mainly bind to in techniques where we are working with proteins which are denatured. But there are also so-called conformational discontinuous epitopes, and look what happens. Here, antibody is binding to amino acids which are brought by protein fold. And these epitopes antibodies are binding to when we work in techniques like immunoprecipitation, cheap Western blot in native conditions. So these epitopes, we have absolutely no possibility to predict in any way. So in Western blots, antibodies are binding mainly to linear epitopes, and in other techniques where protein to be detected is in a native conformation, we have discontinuous epitopes. And when you receive the PDF of this presentation, you will be able to follow these links, which are provided almost on every slide if you would like to obtain more information. And also, you're probably familiar with the terms of poly and monoclonal antibody. By some reason, monoclonal antibodies are somehow perceived to be better or giving less background. I wouldn't say that they are always better. They are definitely not so versatile as polyclonal antibody. And I will explain why. A polyclonal antibody is binding to a pool of several epitopes on a target protein. And these can come from different species, here ordered regarding to the size, how much of the antibody can be obtained from different species if we require larger quantities. And if you remember the linear epitopes, a poly polyclonal, poly means many, will bind to different epitopes on a target protein. So it will be a pool of several epitopes this antibody will bind to. While monoclonal antibody, mono means one, so it will detect only one antibody epitope. So if with time an antibody deteriorates, in case of monoclonal antibody, it will just lose activity, it will stop working. This cannot be predicted. Polyclonal antibody, though, which is a pool of different epitopes to a target protein, may still maintain detection. Maybe not at the top level, but it will be still working. I know that the monoclonal antibody to callose, for example, has lost activity. Which have been, and it is not possible to predict how long an antibody is going to be actually useful, because it is a protein and its stability can actually change over the time. So remember, epitope is 3 to 15 amino acids, and when we are producing an antibody, choice of poly or monoclonal antibody will depend upon our application. In some applications, monoclonal antibodies will be preferred, but for a Western blot, polyclonal antibody will work just fine. Antibody choice also of which one you're choosing for your experiments, it must match species. This is very often overlooked, unfortunately, which can contribute to lack of results because, of course, vendors are not maybe disclosing the exact sequence which was used to elicit an antibody which we want to purchase. But we need to ask. And Agrisera receives several questions of this type per day, which are, I'm working with, for example, Setaria or some other species, and would like to use this antibody. Is it going to work? And it is actually possible to predict. The vendor who has a sequence of antigen used to elicit an antibody is able to predict if an antibody is having a potential to work or not. And please remember that, because it will save you enormous amount of time in the lab, time and frustration. Because if you choose an antibody which does not match your species, of course, there is not going to be a signal. And also you need to keep in mind that with antibodies, it is possible to obtain false positive results. So sometimes in publications, I find that an antibody, Agrisera antibody, is used on a species where is no conservation, but the result is a published band. And if I find it in time, I am contacting the authors. It is also possible to predict if an antibody is going to match an application, because if an antibody was made to a short peptide chosen from the inside of the protein, which is not in a protein fold, which is kind of hidden in some loop, this antibody is not going to be suitable for immunoprecipitation. Remember, we need an antibody either for work on the natured protein, or if we want to use an antibody in native techniques, it needs to be done to a part which will be exposed, and therefore antibody can bind. So it is really crucial that before you use any antibody which you receive from a colleague or which you purchase, you actually do that check. It will really save you a lot of time. When Agrisera is producing antibodies, we are thinking about covering, because in plant science it's a lot of model species, so we are thinking about covering quite many model species, of course, if it is possible. Especially for photosynthetic proteins, which are quite conserved, one can choose a short peptide, which will be found across hundreds of different species. And we call these antibodies global antibodies. Such situation is possible only when the short peptide is perfectly conserved. And therefore, so-called predicted reactivity of a given antibody can be established. You will find it on our product info sheets that we have confirmed reactivity and predicted reactivity, which is coming from sequence analysis. And now, how we are choosing how do I choose the right antibody for my protein? That was one of the questions I have received. Here you can see a sequence, an example of a sequence of a photosynthetic protein, PSB-A. And in this case, we have three scenarios. Look, the peptide which is used to produce this antibody can come either from N-terminal, of course, after signal peptide, or C-terminal. And these will be exposed in the native protein. So, and such antibody can be used also in native techniques. But if it comes from the buried part of some loop, it will not work in a native technique. It will work in the Western blot, in the natured conditions. And oftentimes antibodies are produced to shorter peptides. And the reason for it is that if we're targeting a protein from a family where we have a lot of isoforms from, let's say, 1 to 10, and we want to cover only isoform 1, we need to choose the peptide which is really specific to the protein we aim to detect. In case of this photosynthetic proteins, we wanted to choose peptides which will cover multiple species. And if one is injecting, of course, a protein of certain molecular weight, such an antibody will be a pool covering quite many epitopes. So, of course, peptide antibody will cover less epitopes than an antibody to a whole protein. But there are also limitations with that because the background signal and cross-reactivity can increase if an antibody is made to a whole protein. So it's not always an advantage. Required for so-called predicted reactivity is at least 70% linear sequence identity for an antibody to work on untested species. So please keep in mind that the success of your of your Western blot experiments starts actually with the choice of the antibody. So before you even go to the lab, your species and the antibody need to match. And just a reminder how antibodies are actually visualizing our target proteins. It can be done in this way. So we have a target on the membrane, which is blocked, of course, and the primary antibody binds to the target. And then we have a secondary antibody conjugated with a dye or a fluorophore, which will facilitate visualization of our target protein. So this is one of the possibilities. And there is also a possibility that the primary antibody is directly labeled. And this can shorten your Western blot time because you don't need an incubation with secondary antibody. And this may work well for protein of high or moderate expression. If you work with proteins of low expression, the enhancing effect of a secondary antibody is very valuable. And now we are walking into the Western blot protocol, which, as you know, has quite many steps. So it's a lot of different parameters which can be optimized. And of course, we are aiming for the result like this. But this is a theory. The practice can look a little bit differently. Here I show you examples from 4 different laboratories which received the same antibody. It is to transcription factor, PIF, It is not an easy antibody not an easy protein to detect because it is present in tissue in low amounts. And as you see here, the results are very different. Oh, sorry. From no signal to quite a lot of background. And one of the labs was able actually to optimize the conditions to visualize the target protein. When working with unknown antibody, it is good to include the controls. So not to only assess that target protein binding occurred based on molecular weight, but also have some type of control. In this case, there were mutants and overexpressor. Because it is, as I mentioned, it is very easy to get false positive results. So please see to it that you are using the antibodies which are properly validated. So to be able to actually visualize your target protein, you need to do a little bit of theoretical work, which will help to facilitate the binding between the antibody and the target protein. So here is the checklist. Biochemical properties of your proteins, protein you aim to detect, isoelectric point, its solubility. Is it a membrane protein or is it, what is its expression level? That's very important. Is it low expressed protein or very abundant? Where is it localized, in which organ, which tissue? These are the crucial informations. And also cellular localization can be of importance, especially if protein to be detected is of low abundance. And you may say, well, but how do I know how much of my target protein is in a tissue? There are actually databases. My favorite is the PAX, where you can check on a histogram, you can check estimated amount of target protein in a tissue. It is not, of course, for all species in plant science, but for main model species, you will obtain this information. And as a rule of thumb, if a protein is abundant in less than 10 ppm in a given tissue, we are hesitating to make an antibody because it means, especially if a protein has a cellular localization in cytoplasm, it may be difficult to detect it. Not impossible, but it may be difficult. And there's also ePlant from University of Toronto, which covers quite many model species and which gives us expression level on mRNA level. Of course, mRNA may not completely translate to a protein, but it will give you anyhow an idea of also in which organ you will find the most of your target protein. Sometimes you may need to analyze senescent leaves, sometimes a very root tip. I mean, this is very important when planning your Western blot. Which tissue are you going to process? Here we're coming to sample quality, which is now we are coming to the lab. From a theoretical preparation, we are coming to the lab where we're going to extract the protein. If you would like to use these nice images, they are available on the Agrisera website for free download and use. We have, I don't know which extraction method you're using, but there are different possibilities here depending upon how many samples you have to process. If you're processing multiple samples at the same, I mean, like 10, 20, I would recommend you a bead beater, which is used for DNA, RNA work, but it's also suitable for proteins with a proper optimization of the protocol. You can extract proteins using glass beads of a certain diameter. You can optimize it depending upon what material you're working with. Time-consuming extraction methods are mortar and pestle, and also sonication, which may, in this case, a heat can be generated, excessive heat. So one has to really watch for these parameters. So please keep in mind that your samples need to be of the best quality. And I would not recommend to analyze samples which are several months old, because for some target proteins, especially of those of low expression, there may be a degradation. So lack of signal may be contributed by target protein being degraded. We are preparing an educational poster about protein extraction, which should come out sometime in June. If a protein is of low expression, low abundance in the tissue, I would highly recommend you fractionation. Fractionation to specific organellar fraction. As you can see on this example, there was absolutely no signal in total leaf. While when concentrating, analyzing specific fractions, in this case, the target was present in photosystem 1, so photosystem 1 fraction gave the best signal. And I think quite many antibodies have been discarded at this stage. They were checked only in total cell extract, and due to lack of a signal, the experiments were abandoned. If your protein is localized to nuclei, for example, and it is a transcription factor, I would highly recommend to analyze nuclear fraction. Fractionation is considered to be a laborious technique, but actually there are protocols which can be completed within can be done within a few hours. It's not very lengthy procedures. You have to remember also that when you work with protein extraction, you have to be vigilant, you have to work very fast, you have to be very well organized and do a theoretical analysis first, what buffers you're going to use and what methods. So as you can see here, you can actually save your day by using specific cellular fraction. And protein extraction method, when it comes to buffer composition, can actually matter. Here, there's an example from one of Agrisera test labs, which after adjusting extraction buffer composition, actually obtained the target band. Most commonly used is trees, but it's not so well buffering. Hippies has a better buffering capacity. And also your protein extraction method will depend upon what type of material you are using. Is it having a lot of fat or polyphenols? There are different methods which are available to cope with these substances. As you see on this example, buffer composition may matter in that respect, that addition of 6 molar urea to extraction and loading buffers have actually unfolded the protein and allowed an antibody to bind. What I'm showing here are so-called garden blots. So we have samples from barley, Arabidopsis, and rice. And in a first attempt, a signal was only detected in Arabidopsis, while after addition of urea, it was the target protein was unfolded and accessible for the antibody. This is actually some laboratories I know are using urea in their buffers as a default because when doing the Western blotting in the natured conditions, it will allow to really keep the protein unfolded. So there can be actually difference depending upon which extraction buffer you're using. As I mentioned, using specific organ can have a crucial importance. For example, for so-called pin proteins, using 2 millimeter of a root tip will be very important as this is where the target protein is concentrated. We also need to include, of course, protease inhibitors and in some cases proteasome inhibitors too. There was a lab who was working with an antibody for a year, and there was no success. And desperately, in the last attempt, they included a proteasome inhibitor, and it appeared that the target protein they were aiming to detect was a target of proteasomal degradation. So including the inhibitor really helped to facilitate the detection. And you also need to, if your protein is present in low amounts, you need to optimize your extraction protocol for a good yield. This is important later to be able to load more per well. How should we work with Western blot? Maybe you know the GLP rule and what it means. Good laboratory practice. And good laboratory practice is that we're working clean. You need to clean the whole equipment very well, very thoroughly. Also, working in the cold is very important in case of Western blot. We have calibrated pipettes and we use fresh reagent stock. This will all help us to get consistent result. And we need to follow the protocol. This presentation is based on the questions. So one of the questions was about the formulation of the gel, and I'm showing you here the example of difference in results obtained between tris glycine and tris acetate gel. And this is like a refining step of the Western blot procedure. If one wants to increase the sensitivity or want to get better results, the gel formulation may be of importance. Why using precast gels was one of the questions. Precast gels can actually are prepared in controlled conditions and can give you higher reproducibility. Sometimes, depending upon the temperature, if you're casting gels yourself, there can be differences in how the gel is polymerizing. So if there is a possibility, and if you're using, if you aim to do quantification, then using precast gels may be helpful. But of course, self-casting works very well if we mastered that technique. What should be loaded per well? That was also one of the questions which I received. Protein, chlorophyll, or volume. Recommended is microgram of protein and microgram of chlorophyll, and this is what I hope you're using. I'm terrified to see that volume is used as loading is done on volume. So 200 milligram of tissue is extracted, and then how much protein is loaded per well, nobody knows. And there are reagents which will allow you to measure protein concentration in presence of different components of extraction buffer containing detergent or denaturants and reducing agents and so on. So this is not an excuse to skip the step of estimation of protein concentration. If you're loading on volume, I don't understand how one can compare the samples, the lines in between each other when loading on volume, especially when doing manual protein extraction. There can be a difference between the samples in how well a sample is actually extracted. So I really hope that you're loading your gels based on protein. If you're working with photosynthetic tissues, you can use chlorophyll. How much material should I have? Should it be nanograms or micrograms? It will also depend, of course, with what protein you aim to visualize. If you are working with a recombinant protein and you want to check expression level, then it's nanograms. Otherwise, if you use chemiluminescence, it can be too much of a signal coming up. One can do a titration. So you can do varied protein load per well to find the best signal to noise ratio with least background. Of course, like in this example here, there was the same protein load per well, but the antibody was used in higher dilution, which diluted away the background. So please remember that if you load too much, of course, that can contribute to also increased background. It's a very fine balance here. Should I use 1D gels or 2D gels? It depends what kind of biological questions you are aiming to answer. Just a reminder, one-dimensional gel electrophoresis, then all proteins in a complex mix, which we are loading here to the well, are separated and migrated in a gel, which has, which is often having a different percent, and which means that proteins which are larger will get stuck faster, and those which are smaller will migrate lower down the gel. And please remember that each such band contains hundreds of proteins, right? This is of a similar molecular weight because we're working with a complex mixture which we are analyzing. So this is so-called one dimension, and it has no PI limits. But sometimes we want to know how many proteins and which proteins are actually in this band. And therefore, then we are using a two-dimensional gel electrophoresis. And one can also do Western blotting on 2D gels. It is, however, less common. But it is another way to analyze proteins. It will allow us to by analyzing the spots, we are analyzing hundreds of proteins at the same time. So that's another methodology. Sample denaturation. I'm curious what temperature you are using. You're probably using 100 degrees, 5 minutes. There are different possibilities here. I would not recommend boiling the sample. And the reason for it is that excessive heating can induce aggregation of proteins. And therefore, if a target protein aggregates, it will not enter the gel, and therefore there will be no band which is detected. This is a classical example is membrane protein HATPase, which if sample is heated in 90 degrees and more, it will aggregate, and as a result, there will be no signal in Western blot. So we are using 70 degrees for 5 minutes, which is enough to denature protein sample before loading on a gel. And this is how we are doing our protein transfers. We are using ice-cold buffers, and we're doing it in ice buckets. Of course, it's also possible in a cold room. Please remember that temperature is important to avoid the smiling effect, for example, of your bands when they are visualized. How important are conditions of the transfer? Is it better semi-dry or wet, and so on? You know now it's important to have it in a cold, but look here. It is important also what protein you aim to detect, because for higher molecular weight proteins, they can be longer retained in a gel, so require longer transfer time. Proteins of lower molecular weight require shorter transfer time. They can be more easily blasted through the membrane, because of their low molecular weight, which is actually also sometimes the reason people don't visualize low molecular weight proteins on the Western blot, because they left the membrane. And there's the way to check it and optimize it. And as you see here, actually it would be ideal to have two different membrane sizes in the lab. And when detecting proteins of higher molecular weight, we use 0.45 micrometer membrane, while when working with proteins of lower molecular weight, we use 0.2 micrometer membrane. And also a little bit, we need to think about transfer buffer, because high molecular weight proteins may require more SDS and less methanol. Also, wet tank transfer may work better for proteins of higher molecular weight, but of course there are always exceptions, and I saw good blots done in semi-dry conditions for high molecular weight proteins. But this is a part of optimization of your Western blot conditions to get really good results. Which membrane to use, and as I see it in this blots which I'm troubleshooting, the majority is using PVDF, but it doesn't mean that it is a better membrane. Both membranes work, but sometimes it depends upon target protein antibody combination on which membrane is going to be better. PVDF has another chemistry of binding the proteins. It can be air dried and re-wetted, which is a very good advantage, and has a better protein retention. It's more robust membrane, PVDF. Nitrocellulose is more fragile and has another mechanism of protein binding. And of course, there will be always exceptions, for which protein antibody pair, which membrane will be more optimal. I would never like to say that one is better than the other. Ideally, it would be good to have both in the lab. I have seen that for some antibodies, less background was obtained with, for example, nitrocellulose. We even write it on our product info sheets. Protein transfer type, well, I wonder which one you are using, wet or semi-dry. There are advantages and disadvantages of each of these methods. We have more buffer we need for wet transfer. We have another electrode contact, more direct with semi-dry transfer, and of course the speed is different. And one should consider that wet transfer is less prone to failure. So of course both will work. It just depends upon how skillful an operator is to assemble wet transfer or a semi-dry transfer. You also need to keep in mind when you're using semi-dry and wet transfer about actually cleaning the electrodes. How to efficiently remove air bubbles? Well, what are the air bubbles doing? If your target protein is where the air bubble is trapped, then of course this will influence the transfer of your target protein. So you will get it less. And here, well, we are not doing like that anymore. We are assembling our sandwich directly in the cassette. So moving the sandwich can contribute to unsharp bands. So you should assemble the sandwich actually directly in the cassette. And sometimes we even add one more pad to make the sandwich sitting really tightly. And on the YouTube, we will post a short video showing how we are gently pushing the air bubbles from a membrane. If they are at the very top, we don't bother. You should move the sandwich as little as possible, actually, and make sure that it's tightly sitting in your cassette. We are doing when working out a Western blot protocol, we are actually doing Ponceau or Comasi R250 reversible staining to check how the separation was looking. You see here, after the markers, one saw that something went wrong, probably overheating. So if we see that the membrane after protein transfer looks like this, after Ponceau staining, we will not continue with it. Something went wrong. It's a waste of time to continue further. Both Ponceau and R250 reversible stains are the dyes can be washed from the membrane. We are not doing this check every time. We are just doing it when we are establishing the protocol for a new target protein. What you can also do, which is a very simple and easy check to confirm how well your transfer was working, is to actually stain the gel after the protein has been transferred to the membrane. If the transfer was complete from the gel to the membrane, the gel should be empty, no staining, not like this. And here you can see that there is actually some protein left in the gel. And this was a double sandwich. So all protein from a first gel was transferred, left the gel, and hopefully was captured by a membrane, but it was not the case for a second gel. So since that experiment, we stopped using a double sandwich with two gels. We are using only one, especially when doing quantifications. But this is a very easy check of your gel to just stain it quickly with Kumasi. And if you see any protein left in the range of molecular weight of your target, you should revisit your protein transfer protocol. Probably it was too short. As I mentioned, for low molecular weight proteins, there is a danger that they will be blasted through the membrane. And you can check if your Western blot protocol is too harsh. Then you can put actually one more membrane in your sandwich. And that way, if a transfer was, voltage was too high, then the protein will be blasted through first membrane and retained on the other membrane, on the second membrane, which you can stain with Ponceau or, with Ponceau or with Kumasi. So just checking how your gel is looking and membrane is looking, you can assess the quality of your transfer, because of course the aim is to detect the band. And primary antibody format is actually also important for your success. The antibody can be provided as serum, or it can be provided purified to total immunoglobulin fraction and purified on specific antigen. And here you can see lyophilized antibodies. This is the method which will secure that in case something happens in a transportation, the antibody is safe as lyophilized powder. And also no need of shipping with dry ice is necessary for lyophilized antibodies. However, if you would like to use an antibody for Western blot, a serum format is often enough, but you should keep in mind to check. Some vendors are purifying antibodies only to total immunoglobulin fraction, and then it looks like you're buying a lot of antibody. But in reality, it is still 200 or 500 micrograms will be a total antibody, not your specific antibody. So in serum, we can estimate concentration of your specific antibody. It can be done after the serum is purified. If total immunoglobulin fraction contains only 0.5 to 5% of a specific antibody. This number is calculated after hundreds of specific purifications, which we have done over the last 20 years. And of course, antigen purified antibody, then in the tube, you will have the most of your specific antibody. And here is just a reminder how the purification is done. So we have resin, where we are coupling the antigen, so protein or peptide, which was used to produce the antibody. And then we are passing the serum, and specific antibody will get stuck on the protein or peptide used to produce the antibody, and then elution is usually low pH to obtain a specific antibody fraction. Here will come some exciting questions. How long can the same aliquot of antibodies stored in blocking buffer be used? So, of course, antibodies are expensive, and they often are reused. And there are no golden rules here. Some antibodies can be used only once, and some antibodies can be used 20 times. I know that some of Agrisera antibodies will be used 20 and more times. And I would not recommend it to reuse the antibody for an antibody you are just characterizing or you don't know how it performs. Because if it is a low titer antibody, you will not get a signal in a second reuse round. And this is not an antibody fault, really, because all of the primary antibody bound in the first incubation, so there's nothing left. That antibody cannot be reused. So please keep it in mind that to be careful with reusing the antibodies, and vigilant when you don't get the signal in a second reuse round, which will mean that such antibody is not suitable to be reused. Primary antibody suddenly does not work any longer. What can be the reason for it? Antibodies are proteins, and they have different stability. And there can be different factors which contributed to antibody losing an activity. It can be storage temperature. The freezer maybe was going up and down in temperature. It can be also purity grade, which will influence how long the antibody is going to be stable. Actually, antibodies in serum can be stored often quite much longer than antibodies which are purified, due to stabilizing proteins which are present in serum. Sometimes antibodies require stabilizer like glycerol or BSA, but please do not add glycerol to serum. It's absolutely not necessary. And if an antibody requires a stabilizer, it should be, if it's a catalog antibody, the information should be provided on a product information sheet. And also, sometimes using the same antibody solution multiple times without inhibitor of bacterial growth can contribute to antibody degradation. From our experience, some antibodies can work for 40 years, while the others may lose activity after maybe a couple of years. And as you remember, there is this FC region, the handle, and that one may influence the stability of antigen binding domain. And as the antigen binding domain is different for each antibody, there's unfortunately no way to predict how long the antibody is going to be stable. Everybody has some problems with background in Western blot before proper signal-to-noise ratio is achieved. Here's an interesting question which I have received from one of the participants who had produced an antibody to mitochondrial protein, but there was still a lot of proteins recognized, even on mitochondrial samples, even with purified antibody. So he wanted to somehow poison the antibody to remove the background using a mutant lacking target protein. And this is actually a protocol where we can remove background signal by depletion. So we prepare a membrane with a mutant sample which is lacking target protein. It has all these background bands coming up, but not a target protein. And then we proceed with the incubation of primary antibody as with a regular blot, and then we collect that solution and use it later. So the aim of that treatment is to make antibodies, background antibodies in the antibody preparation to bind to the proteins, other proteins, background proteins, and that way to kind of purify away this background. This is how you can also remove, if you have problems with Rubisco, cross-reactivity to Rubisco. This is how you can remove these antibodies which are detecting also Rubisco. I mean, rabbits are eating a plant-based diet, so oftentimes they actually will have quite many antibodies to Rubisco. And if your target protein is in proximity of Rubisco, of course, it will be making difficulties with the detection. And this protocol is also described on our website if you would like to use it in the future. But background and membrane blocking, please do not use these kind of tweezers like we did here. They should have flat ends, not to damage the membrane. Is blocking temperature sensitive? That was one of the questions. It is depending upon protein and antibody pair. And we are actually using the PVDF membranes, which we are drying after blocking, and they can be used 1 to 2 weeks later. Or without blocking, we store PVDF membranes for several months. So you can stop in your Western blot protocol at certain point and continue later. This is how we are storing our prepared membranes. You need to keep in mind that this is only possible with PVDF membranes. So we have certain sets of samples which we have separated on a gel, transferred to the membrane, and then we're storing these membranes up to 6 months. So your precious samples actually will be safer on the membrane than in a cell extract in a freezer. And you dry them protein side up, and then you can put them in a plastic bag for storage. Also, this kind of treatment will further denature the proteins, because air drying will further unfold the proteins. Some proteins may partially refold during Western blot procedure, and that way they will make binding place for antibody less accessible. So this is a good tip, and we have a whole box filled with these membranes, which we can use with specific samples up to 6 months. And when it comes to blocking, please see here the examples which I prepared for you. There are different blockers, of course. Over the years, I've seen that most efficient with plant samples is 5 to 10% non-fat milk. We have also commercial blockers which perform quite well. We have PVP40, which is non-protein blocker used quite successfully by some laboratories. We have BSA, as you see, lowest down the list, and gelatin. I'm not really fond of it. Some people are mixing non-fat milk with BSA, but I don't think it really matters. And here you can see 2 examples. So in this first example, we have 30-minute blocking, 1% milk, and the last says antibody is worthless. It's so much cross reactions here. And then I ask them to extend the blocking time to 1 hour and increase the blocker to 5%. And then we have blocked unspecific binding places, and the signal to noise ratio is quite good. So in this case, the blocking was done in room temperature, and that was enough for this protein antibody pair. But in some cases, the blocking has to be done overnight. This is this second example here. So the blocking was done in 5% milk overnight, because blocking at room temperature was not enough. So please keep in mind, this is strictly dependent upon your protein antibody pair, and it needs to be empirically tested. Uh, Later on, when you know your antibody and you're aiming for publishable results, you can do a titration like here. So you can use different protein load per well and different combined with different dilution of primary antibody, and then you can see which one gives the cleanest blot. Of course, for strong antibodies, the more protein you load, the more signal you will get, but also the background bands will show up, as you see in this example. 10 microgram of protein per well, an antibody quite concentrated, and then, of course, we have a background signal. So this is one of the nice optimization steps. I got a question also how to reduce background signals while stripping and reprobing. And please keep in mind that the background signal will increase when doing stripping and reprobing because it is a quite harsh protocol and you are actually breaking the bond between antibody and a target, but also you can wash away some target from a membrane, and that can be a reason for increased background. Comparing primary antibodies, does it work? One is working with not fully characterized primary antibody to protein of low expression and compares it to actin. And well, actin is a high abundancy protein, and therefore you cannot compare it with a protein target of low expression, which may require longer time to visualize the signal. And we are coming here to loading controls, which should allow us, of course, I hope that you're going to load based on protein, and therefore you will require some assessment of equal protein load per well. And in plant science, we have different proteins which may be used as a loading controls, and here it is a short overview. However, please keep in mind that you need to choose a protein, a housekeeping protein, which levels will not change in your experimental setup. And for example, actin, in drought stress, the levels of actin will be changing. So that will be not the best loading control for these conditions. So it is quite crucial that you analyze what loading controls are available and if these proteins are actually stable in your experimental conditions, which antibody provider can not really know. When it comes to antibody incubation buffer, I would like to share with you an interesting example. From my experience, most of the labs are using TBST. However, for this protein antibody combination, PBST buffer was giving less background. And you need to also keep in mind that PBS buffers should not be used when you're working with phosphospecific antibodies. This is similar to this result are posted on our blog, on our technical blog. And now we come to the last step of Western blot techniques to the antibody incubation. There are different ways to actually shorten the time of the incubation. And here, yes, you see well, there are 3 primary antibodies incubated at the same time. This is one of the ways to shorten your Western blot protocol. Of course, I wouldn't advise to take unknown antibodies and incubate them together. But if you work with antibodies which are characterized and you know how they are performing, you can actually, if you have limited number of samples you would like to process, then you can actually use several primary antibodies at the same time. But please keep in mind that proteins you aim to detect should be at least 20 kilodaltons apart from each other. Second method of saving time is to incubate everything at once. This is for high titer antibodies, so we are incubating blocker, primary, and secondary antibody at the same time. Yes, it is possible. And it will save you your Western blot time by actually a few hours. But of course, I wouldn't recommend this for an unknown antibody to a protein which is a transcription factor. Here, this is a photosynthetic protein, which is quite abundant, but such tricks can help you to actually shorten your Western blot protocol time. And the third method, we have also possibility to use directly conjugated primary antibodies. So in this case, it is Rubisco antibody, which is conjugated to horseradish peroxidase. And this method works for proteins of high and moderate abundance in the tissue. So the primary antibody can be conjugated to an enzyme or fluorophore, and therefore you will shorten the Western blot time as there is no need for a secondary antibody and washes. And when coming to secondary antibodies, they are done in different hosts, and they are against there is quite a lot of secondary antibodies to choose from. One can be a little bit lost sometimes, but I will explain to you what is important. Here is a reminder how an antibody molecule looks like, and some antibodies are made to this FAB region, and the other ones are made to FC region or to a whole molecule, as it states here. And if you are having a primary antibody made in rabbit, of course, your secondary should be anti-rabbit. And why does it matter which secondary antibody you choose? Actually, they can have different sensitivity. And also, it is important that you use secondary antibodies in high dilution. Agrisera goat anti-rabbit can be used above 1 to 25,000 dilution, 1 hour incubation. You don't need to incubate longer with secondary antibodies. And we are so concentrated on when buying secondary antibodies on the amount of antibody in a vial. If it's 1 milligram or 5 milligrams, but it's actually not that what is important. It is important in what dilution you can use the secondary antibody, because if you can use it in high dilutions, 1 vial will last whole PhD. So it is important that you actually look at the dilutions, recommended dilutions of secondary antibodies. And now, see here, which secondary antibody would you choose for Western blot? We have goat anti-rabbit whole molecule. This is H and L, heavy and light chains of IgG, or this one, which is adsorbed to minimum cross-reactivity to bovine, human, goat, mouse, rat, IgG. Also, think for yourself, which antibody would you choose for your Western blot, and I'll provide you with the answer. Actually, the first one is enough. The second antibody is adsorbed against all these different proteins, and these adsorbed antibodies are especially important to use in immunohistochemical studies, because the antibody preparation is passed through several columns to remove as much of cross-reacting antibodies as possible. But for Western blot, a simple goat anti-rabbit horseradish peroxidate conjugated antibody, if you use ECL detection, will be enough. And please remember, in the future, look at secondary antibody dilution. The price is not really, of course, that is important, but the dilution will tell you for how many assays you can actually use the secondary antibody. And now we are coming to the last steps of the Western blot. We want to visualize our target protein. My favorite detection method is chemiluminescent, and it is still one of the most commonly used methods for protein visualization in Western blot. And here you will see a set of ECL reagents, which you may win. If you have participated in a quiz, you will have a second chance. These reagents are of different sensitivity and they are delivered together as a set. So if one of them is giving too much background, you can wash the membrane and develop with the other. And detection, chemiluminescent detection is facilitated by using horseradish peroxidase conjugated secondary antibodies. And of course, we need CCD camera. And here you have an example of a blot used with the reagents from the previous slides. So ECL super bright and ECL bright. One is for super bright is for detecting extreme low femtogram detection range, and the other one is for proteins of moderate and high expression. But what happened here was that if you apply too sensitive detection reagent on a protein of high expression, this is how it ends up. It will be a background coming up within seconds. So you actually need to adjust sensitivity of your detection reagent to abundance of your target. And please don't overestimate that, because the background bands can be actually coming from using too sensitive detection reagent. And if you have the ECL as a set, then you can just wash the membrane and develop it with less sensitive detection reagent, as in this example. Advantages of chemiluminescent detection is sensitivity. Of course, I know that people working with protein modifications are fond of chemiluminescent detection methods. And disadvantage is that the signal is fading, so it has to be recorded immediately. But this method is very good for analysis of proteins present in low levels in the tissue. Ghost bands, anybody had seen them? They happen now and then, and they are due to, if you wonder why, it's too much of secondary antibody excess, and sometimes antibody mismatch can be reason for it. So reaction goes so fast that you will get the white band instead of a black band. So protein overload combined with too much of the antibodies. And if the blot does not develop properly or something went wrong during documentation, can the ECL incubation be redone? How do you think? Yes, it can. It can be redone within the same day. So you are saved and you do not need to repeat the whole procedure. Second most popular detection method is the fluorescent detection. And here you can see a multiplexing and visualization of two different target proteins on the same blot. Secondary antibodies are conjugated to fluorescent dyes, and of course, a CCD camera is required. Advantage is that this signal is present for months, and the multiplexing can be used with fluorescent detection. Disadvantage is a specific dedicated equipment and lower sensitivity. Sometimes if antibody protein fails with one detection method, actually another detection method can save the day. So because we will be using a different pair between primary and secondary antibody and maybe different sensitivity of the whole system. So if there is an alternative to use, if chemiluminescent doesn't work, one can, if there is a possibility, one can try with fluorescence. The least sensitive detection method is colorimetric detection. It is, I would not recommend it for proteins of low expression. This will not work. It is a very convenient method because we do not need a digital camera, and secondary antibody is conjugated to the enzyme alkaline phosphatase, and the blot can be stored in a lab book. However, it will fade. So it's inexpensive and fast, but please keep in mind that if you are after transcription factor, it may be very difficult to visualize it using colorimetry detection method. It is very fast and very easy to work with, though. And no buffers containing PBS should be used. It's just a reminder here. But you will get all the slides if you would like to. So final results can be obtained using these 3 different detection methods. And for proteins of lower abundance, I would really like to recommend you to use chemiluminescence and fluorescence. For proteins, photosynthetic proteins, and proteins of higher or moderate abundance, chromogenic detection method will do the job. And how to determine molecular weight of a target protein detected on the Western blot? Because to my experience, sometimes the results were misinterpreted due to the lack of molecular weight standards, which were coming directly on during detection. I would recommend you to use the standards which have IgG binding site, which means that when you develop your blot, then standards are visualized at the same time. We're using magic marks, which have a nice molecular weight span, and therefore there will be no hesitation to know what is actual molecular weight of a target protein. The markers with IgG binding site are compatible with all 3 detection methods, and I would like to highly recommend you to include them. In our experiments, if we are using ECL, we are mixing just the colored markers together with magic marks in the same well. So we can see migration of the proteins during gel electrophoresis, and then we can have a nice confirmation of molecular weight once we do the final reaction of protein visualization. This question came, and therefore I'm taking it up here. Epitope tag antibody gave no signal. This is actually a very common problem because there are so many proteins, but it's not enough of antibodies on the market. It is estimated that it's a few million antibodies. Unfortunately, to plant proteins, it will be only a few thousand antibodies. Therefore, tagging specific proteins and going through transgenic organisms is the way to investigate a protein of interest. And sometimes it may happen that the same GFP antibody does not work on another protein expressed in the lab. And why is that? Yes, there can be different reasons for it. And this is what you can try. Consider expression level of a tagged protein. Maybe it's too low. It's beyond the antibody sensitivity. You can increase protein load per well. You can use highly denaturing reagent in your extraction and loading buffer. You can dry in the air PVDF membrane. Now you know, if you dry it protein side up, it will further unfold the target protein. Sometimes if above does not work, you need to use another anti-GFP antibody. So with antibodies to epitope tags, there can be partial refolding of a target protein, which may make a tag not accessible for the antibody to bind. And consider also if antibodies were properly stored. That's actually the point which is sometimes overlooked. And now we are coming to how to get an intense signal from Western blot using a weak antibody. Well, before you deem antibody to be weak, you should go back to the desk and do a theoretical analysis. Did you use the correct sample type? Was it fresh for low expressed proteins? If it is a low expressed protein, did you use fractionation? Maybe you had to load too little protein loaded per well. Were optimal transfer conditions applied, or can something be improved there? Did you use antigen purified antibodies, or did you use serum? Did you use appropriate detection reagent? Did it have enough sensitivity? So all this is called optimization, which I was walking you through today. And I would like to show you an example of optimization before and after. So you see here Western blot protocol optimization, which was done. It was one parameter which was changed. Some of you probably will see it directly from the description here. The only thing which is different between those two blots is protein load per well. So it is not that it is a bad antibody. It is a strong antibody which was used on too much protein loaded in a well. So this is an optimization process, which sometimes can be that easy, and sometimes it may require a little bit more. So I have an advice for beginners: record correct band first before multiplying variations of conditions. This is the most common mistake. And after conducting 10 or 20 blots, I finally get the information. And the change could have been deduced on the beginning, before any of this experimental work has been done. So if you work with an unknown, uncharacterized antibody, for example, newly produced, you don't know how to use it, how it will behave. This is the protocol which we are using. So we are overloading everything. We use a lot of protein per well, and we use also very concentrated primary antibody. And of course, there will be a lot of background, but who cares? The aim is to see if we have a specific band. So, of course, as a control, one should include a mutant overexpressor to have some reference and to be certain that detected band is actually a target protein. So I would really recommend you to try this instead of running blot after blot, adjusting one or two parameters. Of course, you need to first be certain that you're using a correct sample from correct material and that your transfer conditions were effective. I have also an advice for experienced users. And the same protocol does not fit all antibodies, but I'm certain you have realized that. There are certain protein antibody pairs which require, in fact, a change in parameters. So the protocol, which was worked out in a lab for protein of high molecular weight, may not be suitable for a protein of low molecular weight when it comes to, for example, transfer conditions. So please keep this in mind. And if you have still such blots, these are from my extensive collection, then you're welcome to contact Agrisera. I am often troubleshooting antibodies which are not coming from us, and I will be happy to help you. And now concentrate to answer true or false on the questions which are coming. Other antibodies I've bought from Agrisera work. Therefore, this one should work too. Is it true or false? Based on what you have learned today, think and in your mind give the answer. It's false, because each antibody is different. One cannot assume that every antibody is going to work if, for example, we did not read product information sheet. The antibody works for species A, so for sure it is also going to work for species B. Is that true? No, because of course one needs to check if the sequence used to elicit given antibody is also found in a protein which we aim to detect. This will save you a lot of time. The same protocol can be applied for all antibodies. I think you know the answer is false. There need to be modifications. Antibody gives a good signal in Western blot. So therefore, it should also work in other techniques. False. Each technique requires its own validation. So if an antibody is messy in Western blot, it doesn't mean it's not going to work well in immunohistochemistry. It's different fixation method, and antigen is exposed in a different way. So remember, please remember, one method, one validation. We cannot compare validations in between and draw conclusions. And on our technical blog, you will find a lot of interesting information. If you would like to, I am posting information which comes from my troubleshooting. So there may be something new you can learn there. And also, I would like um, to make you sensitive for how you are actually citing antibodies in your publications when you're writing a paper. Unfortunately, there is no worldwide standard, which means that over half of antibodies used in research cannot be properly identified. Without product number and supplier, you're not able to identify which GFP antibody have been used in the work. And this, therefore, please try to include at least antibody name, product number, and supplier. This is really very important. And that way we can altogether help to increase reproducibility. And on our free resources, you will find educational posters. The theme is photosynthesis, epigenetics, and this year it will be protein extraction. I would like to also tell you about the global plant science events calendar, which contains over 250 events every year specifically for plant science. It's not only conferences, it's also workshops, it's also online events. And the calendar has been run for 5 years, and it is a mutual effort of American Society of Plant Biology and Agrisera. And if you look for free images of model species or different pathways, you will also find them on our website. And if you would like to be receiving a newsletter, please let me know about it. And as surprise also, we have an offer of over 100 free antibodies. There is no limit of provided antibodies. If antibodies which you choose and your experimental setup, it means species and application fit together. So following that link, you can check the offer. There's also some epitope tag antibodies, as well as antibodies to different plant and algal proteins. And for customers, for researchers from Australia, please remember that we have Sapphire as our distributor in Australia, who will help you to import the antibodies. And actually, this Sapphire is the company which we collaborate longest with. They have been our distributor almost from the beginning. And for researchers from rest of the world, if you have any questions, here are the contact details. So thank you that you wanted to join today. And if you have any questions, please ask them or write them in the chat. We have the time for that.
Excuse me, Joanna. I think Gulshan has a question regarding the reusing of the primary antibody.
Oh, yeah.
Yes, sorry,
Yes,
just mentioning
thank
that.
you. I didn't see it.
That's okay.
Can I reuse the primary antibody after using it at room temperature? And also how to deal with ghost band? Very good question. Okay. Some primary antibodies can be reused, but not all of them. And you need to empirically test it. So when you have like 5 or 10 milliliters of your primary incubation buffer, after you incubate it with a blot, you pour it to a sterile Falcon tube, and then you store it frozen until the next blot. And you need to, I know some researchers put marks on the tube how many times antibody was reused, but please keep in mind, not all antibodies can be reused. So it may be that in a second round, in a second blot, you will not get any band. It's not really a good procedure, but I know it has been, it is used quite commonly because antibodies are expensive. And how to deal with ghost bands. You need to load less protein. You need to use more diluted primary antibody. This is how you can get rid of ghost bands. And if you would like to, I can take a look at your blots also. So you are welcome to send them to me. I'll be happy to help you. Okay, do we have some more? I see that there's maybe some more question coming. I'm having troubles with detection. I constantly get very blurry background on the whole blot. Okay. My first question would be, do you use constant volume, incubation volume? So you need to, if it's written 10 milliliter in a protocol, you need to use 10 milliliter. You need to work exactly as it is written in the protocol and follow everything to the minute. Volume, time, use gloves, and clean all containers. They need to be clinically clean. Otherwise, they may be a background coming up. It can also be not enough blocking. So if you send me the blot, I will be I may have some other ideas. Also, when I troubleshoot, I always want to see whole membrane because from description, it is often not enough. So it is like in a saying, the picture tells more than 1,000 words. Is it possible to store nitrocellulose membrane similar to? No, it isn't. Nitrocellulose is more fragile, unfortunately. So it is PVDF membrane which can be stored like I have described to you. It is a very good and robust membrane. So maybe if you would like to store it, then you should consider switching to PVDF. PVDF membrane, of course, will require activation with methanol before it can be used after it has been stored dried. Constant volume for the incubation with primary antibody and secondary, yes. I should be able, or shall I say, yeah, you can please send me the picture of your blot and I will help you to get rid of the background. So is there any more questions? If you don't have more questions and you would like to receive certificate of participation, here is my email address, joanna@agrisera.com. And also, I would like to encourage you to actually do the quiz. Therefore, I will keep it open until tomorrow. So if there are no more questions right now, thank you for your attention, and you're always welcome to contact me with your Western blot issues. And if you would like to have materials after this after this workshop, like recording, certificate, slides, please contact me by email. And for those of you who are from Australia, it will be Kathy who is going to follow up. So thanks a lot that you took your time in the afternoon, and I hope it was useful. And you are, of course, welcome to share this material with your lab members, and you are always welcome to come back and ask questions about antibodies and antibody techniques. So thank you very much and good luck with your blots.